Good evening, everyone. My name is Adrian Amendano. I'm the Stewardship Programs Manager here at Still Moon Art Society, and I'm here to welcome you tonight to the November edition of the Shedding Light Talk series. And tonight we have a very special guest, Amy. I'm gonna not pronounce your last name. I'm gonna let you do that because I want to respect your name. So we have Amy from the Salmon Coast Field Station, and just a couple of housekeeping things. First thing I want to do is a land acknowledgement. So tonight I'm connecting from the traditional and unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations, and all the work that Still Moon does is on these traditional territories and lands, and it's with my greatest honor that I've had the immense privilege to grow up in this area and to learn so much from so many people and have such wonderful experiences, and I'm definitely very much aware of the context of how that has come about, and it's really important for us tonight to keep that in our minds and our hearts as we continue striving forward to a more equitable place for everyone, especially our Indigenous peoples all across Turtle Island. And so thank you so much for being here tonight. And I'll pass it on to you, Amy. Thank you so much for being here, and we're looking forward to your talk.
Thanks so much for inviting me, Adrian. It's really a pleasure to be able to connect with Still Moon Arts and learn a little bit more about you and your community, and to share the work that's happening at Salmon Coast. My name is Amy Kamarainen, and I have been working with Salmon Coast for about 3 years now. And I'm excited to be here and share what we've been up to. So Keegan, could you go to the next slide? First thing I wanted to do was put out a plug for the fact that we are actually hiring right now. We are looking for a maintenance and facilities manager as well as a station coordinator. So if you, after seeing this talk, are interested in those possibilities, certainly reach out. Here's a webpage where you can find out more information about those jobs. But we're excited to expand our team in this way. And so hopefully you can share that with any folks you know who might be good fits for these positions. I can obviously tell you more if anyone wants more information. Next. Salmon Coast Field Station is on Musqueam territory in the northern part of Vancouver Island region. And so we're located on Guilford Island, and just feel very grateful to be able to do the work we do, in many cases in collaboration with folks from the Musqueam First Nation. Next. So our field station is remote off-grid boat access only. Keegan, you can press next 3 more times to put the bullets up on this slide. And our mission is to lead and facilitate research to help understand and conserve BC's coastal ecosystems, and also to offer educational opportunities for all of our communities, whether that be the local folks who live in and around us, or in this kind of format where I can reach out to folks who are just generally interested in the area. Our third mission is that we try to model sustainable living in those ecosystems and communities, and being off-grid and remote kind of gives us a unique perspective on that in terms of needing to create our own energy and maintain our own water supply, and think about where our food is coming from, because we have to go a long way to get it. It's about an 1.5-hour boat ride to the nearest town, which we go to Port McNeill on Vancouver Island to access most of the usual amenities of a town. Next. This area is, of course, also home to these beautiful rivers that have historically been rich habitat for salmon, and there've been this is just one example. This is a river called the Viner River. Next. And our history is tied to this woman who is Alexandra Morton. She settled at the site that is now the Salmon Coast Field Station, and it was her home before we were there. She started living there. She was drawn there by her research on orca, and through researching the orca, she started paying attention to the salmon. And as she paid closer and closer attention to the salmon, she started noticing some strange things. This was sort of in the late 90s, and in the early 2000s, she raised an alarm bell. You can press the next button when she found just the state of the juvenile salmon in this area. This is sort of like the first time she really put out this public scientific study showing the effects of the sea lice in this particular region. And it really was something that catalyzed her to try to figure out what was going on with the fish in the area, and to invite other researchers to her home to help her study what was happening in the region. So really, like, the whole start of the field station came out of this need to figure out what was going on, and out of Alex basically reaching out to anyone who would listen to come and help her figure it out. And so she invited people to her home in the summers to do research, and she said she would feed people if they worked with her on the research that she was trying to do. So that's how things sort of started at the field station. And next. And I'm sure that a number of you have sort of are aware, familiar with the like iconic life cycle of salmon, but it's a really important part of the story tonight, because you know, as salmon go into fresh water to spawn and lay their eggs, and those eggs hatch, and the young migrate out to the ocean, there's, you know, of course, both a freshwater phase and a marine phase. And one of the things that I want to talk about today is the sea lice that we could see on the surface of the juvenile salmon. These sea lice are ectoparasites. They're parasites that feed off of the surface of the juvenile fish. And next, and in a normal, like in a natural system where the adult salmon are coming back to the streams in the fall, the sea lice don't persist, or they don't hold on to the fish in fresh water. They fall off the fish. And so in sort of a natural system, when the adults are coming in the fall and the juveniles are emerging during the spring, there's not a lot of opportunity for those sea lice to go from the adults to the juveniles. So it's sort of like the juveniles are typically safe from the influence of these sea lice. But in the waters in the Broughton Archipelago, you could say next slide. In the 80s, a number of fish farms started in the area, and this really became a key feature that Alex started paying attention to. And you can press next. Because of course, at these really the high densities of fish that are kept in fish farms, it becomes a breeding ground for these parasites. And if you go to the next slide, you can see how this kind of impacts this life cycle of the salmon. So when fish farms are in the waters near the migration routes of wild salmon, the parasites that are already on some of the adults can basically infect the fish that are in the fish farms. And then in the spring, when the young juvenile salmon are migrating out of the rivers, they can get reinfected by the sea lice that are on the fish in the fish farms. So this dynamic is something that sort of really became something to pay attention to. And if you press next, basically became something that the folks at Salmon Coast Field Station have been working on over the last 20 years, been trying to unravel what is the impact of the fact that these fish farms can act as refugia or resting places for these parasites as they interact with the normal migration pattern of the salmon. So folks at the research station have basically been studying wild salmon using consistent methods over the last 20 years, over 20 years now. And what we do is, up in the corner, you can see people have a beach seine. So it's a big net that we use to collect a large school of juvenile salmon. And once we get them in the net, we transfer them to small buckets on our boat, and we measure them, and using a hand lens, we are able to identify the lice that are on the fish. And there are two primary species of sea lice that we're interested in. One that's specific to salmon, and then one that's more of a generalist and can affect basically a lot of different fish in the area. So we've been paying attention to the dynamics of how fish farms are amplifying the signal of sea lice over the last 20 years. So I want to tell you a little bit more about what we've learned over that period. So next. So things we've learned so far through conducting this research over the last 20 years is that the infection pressure for sea lice is 25% or 25 times higher near fish farms than the ambient infection pressure. So like we know that sea lice are sort of a natural part of this system. They're a natural part. They're on adult fish regardless, but the numbers that we see can be 25 times higher than they would naturally be when there are fish farms involved. The second point that we've been able to learn through the research we've done over the last years has been that this infection pressure amplification that we see can extend up to 30 kilometers from the site of one of the fish farms. So that's pretty incredible. Just in these systems that are dominated by tides and currents, the sea lice can move pretty large distances in their floating stage. So part of their life cycle is part where they actually are floating rather than attached to the fish. Okay, next. And so I just wanted to kind of say, okay, so yes, sea lice are more prevalent when there's fish farms around, but what's the big deal? How bad are they really for the juvenile fish? Here you can see on the top is a healthy juvenile salmon, and then on the bottom is one that's got a number of sea lice on it. And some things that you can notice is the one on the bottom has blotches, sort of lesions that from where the parasites were previously. So you can see some parasites on their surface, but you can also see some of the sort of bruises and blotches from where they were before this as well. And one thing about the juveniles that makes them particularly susceptible to the sea lice is that they don't have their scales aren't fully formed, and so they adult fish have scales that protect their entire sides, but the juveniles don't have that yet, and so are really quite strongly affected by these. We can go to the next. So the sea lice feed on the skin and mucus and blood of the juveniles, can lead to the scarring and the lesions that you're able to see. And I'll talk a little bit more about the ecological effects on some of the next slides, but it can also lead directly to the mortality or the death of juvenile fish. Basically, when you look at one of the, like, to picture like what this might be like for the fish, we try to think about like the size of those parasites compared to our own bodies. And one of my colleagues likes to say it's almost like you have squirrels stuck to you, like this, like parasites that are about the size of a squirrel stuck on your own body. And you can imagine like how distracting or how disturbing that would be to you. Go to the next slide. And this just shows, that given what we know about how the fish farms can amplify the signal of these sea lice, this is a slide that shows the distribution of fish farms around the coast of British Columbia. And so this, Alex really raised the alarm bell in our local region, but this is something that's a problem all around the region and the province and the coast. Next. This is showing more of a zoom in on our little area and showing the prevalence of fish farms in this area. So, when Alex noticed this problem and started paying attention to it, this has been what the context was, how many fish farms there were surrounding the island that we're located on. Next. And one study that came out of our collaborators has been this comparison of, well, okay, how can we tell if these lice are actually having an effect on the fish and their populations? So they did a comparison thinking about the regions shown in pink, which are strongly affected by fish farms, and the regions in orange, green, which at the time didn't have any fish farm influence. And looking at the different trajectories of the population growth for these for pink salmon in these two regions, they were able to use modeling to show that before fish farms were present in the area, basically the population growth rate was equivalent in the two areas. But of course, after the fish farms presence in the southern region, the growth rate was very, very much affected by the fish farms. Next. And you can press next a couple more times. Yep, that's great. And so they've published an article about this in Science, which is one of the leading scientific journals, and just noted, or one of the outcomes of this study was a prediction that if nothing was done, 99% of there'd be a 99% collapse in the pink salmon populations within 4 generations of the fish lives. So 8 years, and that was published in 2007. So here we are, more than 8 years later. So luckily something was done in the interim, but it was a really sort of dire situation at that point in time. So if you go to next, we can and next, and next. Thanks. So just this is a graph showing the data we've collected over time, over that 20-year time, well, up to 2014, this particular slide is showing. But starting in 2001 was when Alex really started doing this field work to monitor the number of sea lice on wild juvenile salmon. And basically, one of the findings was that about an infection rate above about 3 per fish could affect the mortality. And we can see that basically after the alarm bells were raised, they were able to bring the number of lice on the juvenile fish down quite a bit from the original, from like the initial numbers they were documenting in the early 2000s. They were able to bring them down below about 2 lice per fish. And so this just shows that like with some of the management techniques they were using at the time, they were able to effectively control the sea lice on these farms, and they were doing that through a chemical treatment that was added to the feed of the fish farms called, they refer to it as slice, and amamectin benzoate, and this chemical does effectively, it's basically a pesticide that affects the parasites, but what has been happening in the interim is that they basically, the sea lice have basically developed a resistance to that chemical. Next. So the other interesting thing, in addition to like the sea lice affecting juvenile salmon themselves, like individual salmon and the population size and population trajectory of the pink salmon, another interesting dynamic that folks at the station were able to identify is that, of course, when you have these parasites on you, you're as a small salmon, your behavior changes. So the salmon that juvenile salmon are normally school, and schooling is an effective way for them to avoid predation and to safety in numbers. But when there are sea lice on the sides of the fish, they often do sort of funky kind of swimming patterns, and it leads to ineffective schooling. And so as any fisherman knows, like when you have like a flasher in the water, it's meant to mimic a school of fish and then one lagging fish, because the lagging fish is the one the predator is always going to go for. And so when you're in a schooling situation, like any kind of abnormal or kind of movement is something that will attract a predator to attack, and will and what we were able to show through both modeling and through experimentation in the field is that this in fact was having an effect on the interactions among the different species in the system, because the pink salmon were more strongly affected by the sea lice in terms of their behavior, and coho salmon, which actually are predator of both pink and chum salmon, were more likely to prey on the pink salmon when they had sea lice on them. So it's just an interesting, once you start to dig deeper and deeper into how these sea lice are affecting the salmon, the story gets more and more interesting and more complicated in terms of, we're not just talking about salmon in general. Of course, the salmon, this demonstrates that all the different species of salmon have different roles in the ecosystem, and that they, the way that they interact has developed over a long history of evolution, and these sea lice are having an impact on how they're interacting now in the waters today. Okay, next. So as a summary of like what research at the station, some of the big take homes for research at the station has shown, once again, that infection pressure is higher in with farms in the water, that impact can extend up to 30 kilometers, and that high parasite loads can have lethal effects on the juvenile salmon, and that even at low levels of infection, you can have impacts on the behavior of the fish, including changes in the schooling patterns and susceptibility to predation. So that's just sort of a, like, we know this, like this is stuff that we have learned, and through a lot of hard work and a lot of research over the last 20 years, Salmon Coast has basically shared all of this information out through scientific papers and out to the public. But one of the interesting things that's happening these days is the fish farms are actually being ousted from the waters near Salmon Coast. But in some cases, we're seeing that the operations are actually moving to other places, like in particular the west coast of Vancouver Island. And so it's just sort of what's happening now is like, it seems like almost sometimes people are forgetting that we already know these things about how sea lice and fish farms can affect the ecology of wild salmon. So one goal of Salmon Coast is to just help spread the word and to communicate this science in more different and creative ways. And that's why I was really excited to like hear from Adrian, because I feel like one way our group has had really good success in the past is like working with artists and other folks who are creative and have interesting thoughts on like how to communicate these kind of complicated ideas to different audiences. So it's something I would love to like hear more from any of you about. Anyway, next.
I have a question for you in regards to that, Amy.
Sure.
It's very interesting.
Yeah. And you can press next. Do you want to ask me now, or did you want to?
Oh, I'll save it for the Q&A, but
okay.
Sounds good. As a comment.
Sounds good. Because I'd be happy to do it either way. But anyway, I can keep going. Yeah, and so I guess this is showing the location of Salmon Coast in this context of the lots of fish farms, and the blue lines are showing sort of some of the dominant migration routes for the salmon that pass by. And if you go to next, this shows over the last 20 years, we've been monitoring consistently at these 3 locations that are shown by the 3 stars so that we can better understand what the impact is of these fish farms on the salmon in the region. Next. And again, this is showing some more action shots of how we go about doing this. We go out in the spring of each year because of course the young salmon are migrating out from the rivers between about middle of March to about the end of June in our area. And in our waters, the primary species that we're capturing tend to be pink and chum. So that's what we're getting most of out of the rivers in our region. This is just showing sort of some of the process of collecting them with the large net, and then we put them in these Ziploc bags so that we can look at them really carefully and count the number of lice they have and what life stage the lice are at. And the important thing about our approach and our process is that, and that was something that we pioneered at Salmon Coast, is the process of being able to count the lice on the fish without killing the fish. So we put them in a Ziploc bag just for a few minutes, or not even a few minutes, like a minute, and count the lice, and then we release them into the water right at the same location where we caught them, so they can continue on their migration and make it out to the ocean. They have so many different challenges along the way of their migration that we don't want to be one of the challenges in terms of taking them out and killing them if we can and all avoid it. But there are other folks who have been doing similar monitoring in the region who do use lethal methods for collecting the fish, and they try to argue that it's not, like that it's more accurate if you can take them back to the lab in order to examine them under a microscope. But we've been able to show like that our approaches are consistent and accurate and give us good data and keep the fish in the water. So we're proud of having figured out a way to do that and still do science without killing anything. So anyway, that's one thing I wanted to point out. Next. And so over the last 20 years, this is just a graph showing two kind of views of the data. The first one on the left shows the prevalence. So that is showing us how many, like if you caught 100 fish, how many of them would have at least one louse on them. So for example, like this past year, when we caught fish, it was almost 50% of them, or about 50% of them had at least one louse on them. And then the second graph B, is showing how many lice there were per fish. So you can see that over since about 2006, it's been hovering between about like 1 and 2. But before that, it was significantly more than that. And even recently in other regions, Alex has been doing work all over the coast to stay in touch with different groups who are observing juvenile salmon all over BC coast, and are like even right now they're seeing different areas that have 10 to 20 to even more lice per fish, and so we've kind of in the Broughton have been able to see action that has brought these numbers down, but it's like I said. It's not, it's something that others in other regions of the coast need to be aware of and be paying attention to in their areas. Next. And next. And so one of the really exciting things that's been happening recently is that you might have heard about in about 2018, a number of the First Nations in this region occupied some of the farms, and then well, it was actually prior to 2018 that happened. And then in 2018, they were able to engage in what was called nation-to-nation negotiations around the presence of fish farms and how fish farms were being managed in the area. And through that process and in working with the fish farms, even in this region, they came to an agreement to remove the majority of fish farms from this region by 2022, and with the rest of them being subject to monitoring and to an ultimate agreement or an ultimate decision about whether they would be removed as well. So we're kind of in the middle of a process. And the group that's organizing it we've been working with them in some ways, and it's just been a wonderful collaboration to be able to support a First Nations led group who's been really doing amazing work to monitor the fish in this region and to start some restoration efforts as well. So it's really exciting, but it's also we still feel like we're on our toes because not all the farms are out yet, and we're still seeing lice on the fish in this region, because as you can see, the migration route goes past some of the hot spots before going downstream, and so the out migrating fish can pick up lice basically at those upper reaches, and then those lice are still in the system up to 30 kilometers away. So next. This is just showing that hotspot again of the different farms all over the coast, and I wanted to just emphasize that Salmon Coast has learned a lot by paying close attention to what's happening in our waters, but part of what we want to do now is like help others be aware of what has happened to in our waters and how that played out, and helping others to have the resources and any training that people might need to pay attention to this in their own waters, because there's different levels of pressure from fish farms in different areas of the coast at this time. Okay, next. So one way we're doing this, we're really excited that next year we're planning to offer a sea lice training workshop, which is meant to be an opportunity for community groups or folks who want to know more about the salmon in their regions to be able to learn the techniques of monitoring sea lice on juvenile salmon in their own waters. So we're going to be doing a sea lice training workshop in collaboration with Cedar Coast and Sea to Cedar. Cedar Coast is based in near Tofino, and that's where we'll actually be doing the workshop. So if any of you can join us in Tofino or on actually Vargas Island off the coast of Tofino next May, we'd love to help you learn more about sea licing. Next. And another project, so that's kind of my whole spiel about sea lice specifically. And our field station has focused on sea lice as a main research point over the years. But there's been a lot of other projects that have come through the station, and a lot of other ways that we've better understood the ecosystem around us and the salmon around us. And so I wanted to feature a few of the other projects that have done really good work in the region as well. And one of them was a study that was conducted about 3 years ago, where they assessed just how the wild salmon populations were generally doing in our region of British Columbia. And the interesting thing about this study was it looked at not only the abundance, but the resilience of the populations, because as some of you might know, like salmon, often when stressors are removed from their populations, they do have an amazing capacity to rebound, and to that resilience is just amazing. So this study looked at both abundance and resilience. I thought that was really, and you can find more information about it on our website. Next. One aspect that came out of that particular study was just that historically, the Department of Fisheries and Oceans has been tasked with monitoring the ecosystems and paying attention to how the salmon are doing. And what we found in this process of collecting data for the study I just mentioned is just how much the effort toward collecting data has gone down over the last 50 years. Back in the 1960s, the DFO was monitoring 50 to 60 streams in our region, and counting how many salmon were coming back to those streams every year. And in the last 5 to 10, 15 years, it's been on the order of like between 5 to 20 streams instead. And so this is just one of those things where we can't know what's happening if we're not collecting the information and the data to understand the system. And just with the levels of funding and the support for monitoring, there just has been this degradation of like what we can know about the ecosystem due to just basically degrading funding. So it's just something to pay attention to, and to be kind of getting at our decision makers and our folks who are in charge of the purse strings to be putting effort and effort towards this monitoring. And I mean, it's been up to folks like us, like at these small research stations, to pick up the slack and to try to be out there monitoring and paying attention to what's happening in the ecosystems. And there's a lot that all of us can do to be a part of that. Next. Another exciting aspect of work that we've been working on recently has been being able to take our idea of like not doing any harm to salmon to another level, where we've been playing around with a few different techniques that rely on sort of cutting edge molecular techniques, including one called eDNA and one called one that's more genomics. And eDNA stands for environmental DNA. And it's an approach where here we have Dylan Shea, is a graduate student who's spent time at the field station, and he collects water samples. And from those water samples, he can actually look at like small fragments of DNA that's in the water, and be able to tell both what species of fish have been in that water recently, and also what pathogens are in that water. So he can look at virus and bacteria information in those waters. And he's been looking at, like, we know that sea lice are an aspect of salmon health that's strongly affected by the presence of fish farms. And he's also been able to show that, like, other forms of disease, other types of diseases are also more prevalent near fish farms, which is probably not surprising, but being able to quantify, being able to actually measure that impact is just something that's been a new avenue for research for our group, and an exciting way that we can actually monitor the environment without ever actually catching a fish. We can just do it by catching the DNA that they leave behind as they swim through the water. So that's kind of a neat way to do it. And then the other neat aspect of this overall approach is this a genomics aspect, where the genomics aspect does require that we catch the fish and that we take a small, like tiny, we usually do a fin clip, just a tiny amount of tissue that we can take back to the lab. And when you do that, you can actually analyze that tiny amount of tissue to see what stressors or what kind of situation the fish was involved in right before you caught it. So when the fish is swimming through the water, the genes in its body are being either upregulated or downregulated based on the stressors and the different things it's being exposed to. So the fish, like, we can take that tissue sample, and if we analyze it, the yellow and purple here are showing a comparison of, like, what a fish's genetic profile would look like if it was in cold water versus warm water, or if it's exposed to low oxygen conditions or normal oxygen conditions. So you can see, like, basically the top part of the purple and yellow graph is showing one condition, and then the bottom is showing the other condition. So you can see how different the gene expression looks in these two situations. And so we can use this genomic technique to kind of tell let the salmon tell us what kinds of stressors they've been exposed to. And so it's really exciting because the genomics lab we're working with, it's Christy Miller's lab. She's part of the Department of Fisheries and Oceans at the lab in Nanaimo. And through her lab, she can analyze exposure to stressors, like I was mentioning, temperature and oxygen, but she can also measure a whole suite of different viral and bacterial pathogens as well. So it's also a really exciting new way that we're using cutting edge techniques to help understand salmon health in the region. Next. And some recent work has shown that using, again, genomic techniques, being able to really track based on genetic sequencing where particular viruses are coming from. And so a recent study that was just published this past year was able to demonstrate that the genetic sequences we find on the coast of British Columbia can be traced to the same genetic strain of the PRV virus that is associated with aquaculture in Norway. And the aquaculture companies that are on the BC coast are primarily Norwegian owned, and they get their stock from Norway. So you can see how, like, the actual viruses are spreading through aquaculture, globalized aquaculture trade to British Columbia, and then now are affecting wild fish on our coast. So this is something that we're just able to unravel recently with these genomic techniques. Next. So that kind of wraps up my kind of cutting edge molecular genomics techniques segment. And another project I wanted to, like, tell you guys about and let you know we're working on is one that we've been calling Where Are They Now? This has been a really fun project to work on, and it's more of a communications project than a scientific project, but it's been that now that Salmon Coast has been around for 20 years, we have recognized that like the folks who came through Salmon Coast early in the years are now doing all this awesome stuff. They're in roles with the DFO or with different nonprofit organizations or with Cedar Coast, some of our collaborators now. And so, some folks were like kind of intimately tied to still and still collaborating with, but then there are other folks that have gone on to do really awesome stuff that we're just curious about. And so we ended up reaching out to a number of folks that spent time at Salmon Coast and, kind of were key players in our development, and just asked them what they're up to now. And we created a podcast series that you can check out. And it's really neat because, I've been talking a lot about research today, but Salmon Coast attracts people from a number of different walks of life. And we're really open to like folks who have any sort of community-based interest in understanding the place better. And so, there have been people who come through who are artists, who are storytellers, who are, interested in different aspects of science. And so we're trying to collect stories from all the people from all those different aspects of folks who've been attracted to come to Salmon Coast. So I encourage you to check out our podcast. We're in the process of like releasing them now. So I'll be releasing one actually tomorrow. But yeah, you should just stay in touch with us and watch as our series unfolds. And next. And yeah, then I just wanted to kind of put a plug in for other ways you can get involved with us. Like I mentioned earlier, we do have ways we're hiring right now, but in addition to jobs with us, there are other ways that you can be involved with Salmon Coast. Every summer we take volunteers. Well, with COVID, it was a little bit harder to do, but we did still have volunteers come during the season and used a quarantine system to make sure everyone was safe. But we do take volunteers every year to help with the research and with other aspects of the station, with our gardens, with our maintenance, with everything that's going on. And so if you're interested in volunteering and spending time up there, we are open to that. Also, like I was just mentioning, we are open to being a hub for researchers and for creative people and curious people to come and learn and together. And it's just a really generative place where we usually have between 10 and up to 20 people there at any given time during the summers. And you can apply to like if you have a research idea or a project idea, you can basically bring your ideas to us and let us know what you're wanting to work on. And, we can often work together to make something happen. Yeah, and so there's lots of ways to kind of get involved. So that's kind of the different ways that Salmon Coast has different things we've been working on, ways to get involved specifically with us. I also wanted to share with you a couple of other citizen science projects that we participate in that you could also do in your own local neighborhood, so that if you can't come and hang out with us, you can always like still pay attention to your ecosystem in these other ways. So if you go to the next slide, one of the projects we've been collaborating with recently is one where through the multi-agency Rocky Intertidal network. So if you go to their website, you can get sort of observation sheets that you can use. Any individual person can just make observations in association with this project, but it's to help monitor the spread of sea star wasting syndrome, which maybe you guys are familiar with, but it's been something that's been affecting sea stars all over the West Coast. And this network is trying to just understand the distribution of the effects of this syndrome and like how different areas are recovering from once they're affected by the syndrome. So it's something that if you are anywhere on the coast, you can just go and do this and contribute if you want to. Next. The other example I wanted to share was the BC Citation Sightings Network. It's always so exciting to see a whale, and luckily, near Salmon Coast, we actually see them fairly frequently, and so we get to contribute to this app a lot. but it's a really cool, it's an app that you can download on your mobile phone. And so if you ever happen to see a whale, you can just report it wherever you are, whenever you are. And it's neat because it's just really easy to use. And I don't know, it's fun. So I would highly recommend that even if you're like on the ferry, you can just like be on the lookout for whales and use this app to report them. Next. That's all I wanted to share with you guys today. I feel like I went really fast through this stuff, but I hope it was useful or interesting to you. And I'm just really excited to connect with you guys. So I'd love to kick off the question and answer.
Thank you so much, Amy. That was absolutely wonderful. Yeah, thank you so much for joining us. It was really, really amazing to know all of what is going on. And yeah, I'm super excited for the Q&A. And this is just a wrap up for our YouTube upload. So thank you so much, everyone, for joining. We'll be right back with their Q&A. Thank you so much.